Yeah. There you go. I'll have to, yeah, I know. No, I don't want to sit. I don't want to. Thank you very much. Thank you. Can I use that as it is now? Yeah, that's all right. Huh? Don't put it in my hair. I mean. Oh, you're going through this thing again? Oh, God.
Yeah, I know.
Oh,
what should
God.
I say? He's great.
Huh?
I just say he's great. Let's
Who?
start the talk. You.
No. He's great. What is this fucking flowers for?
Huh?
Yeah, you got to pull his leg a little bit. Are you going to pull his leg a little bit?
He's going to be better at it.
Yeah. He's, yeah. Yeah, he's.
You have the darts, which I don't have.
So you want me to sit there, right?
You can either sit there or you can stand here.
Stand here?
Yeah.
Okay. Whatever is easier. Maybe up so that they can look at me, you know? Yeah.
You want to do it this way?
I'll get up there.
Yeah.
Okay, so you want me to sit?
Yeah.
I will sit.
So, good afternoon and a very warm welcome to all of you. It's an absolute delight to actually welcome an audience that comprises of students, research scholars, and faculty across multiple departments in this university. So your collective presence here actually identifies the true spirit of interdisciplinary research at the University of Calcutta. We are also, we would like to welcome our guests from neighboring institutes and universities who took their time out to come here. It gives me a profound privilege and honor, it's a profound privilege and honor to welcome our distinguished guest and speaker for today, Professor Spyros Artavanis-Tsakonas. He is a leading figure in developmental biology and neuroscience, and he's globally recognized for his foundational work on notch signaling. We are actually, before we begin with that, I would like to first thank the Indian Academy of Sciences for giving us the opportunity to organize this event. For this, I would also like to thank Dr. Sharojit Polle and Professor Kaushik Biswas from Bose Institute for connecting us to them. We truly appreciate your collaboration and support. We are also thankful to the West Bengal Academy of Science and Technology because they have given us the support in terms of broadcasting this program live on YouTube, on the Academy's YouTube channel. And so we have been able to reach a much wider audience. Because when we first announced this talk, we had an overwhelming response beyond the capacities of this hall. So the audience that you see here is only half the online, offline audience. There's another room where we are projecting this simultaneously, where the rest of the audience is actually sitting. You can't see them, they can see you. And apart from that, it's also happening on YouTube right now. I would also like to take a moment to thank the co-organizers, Professor Rajat Banerjee, who's the head of the Department of Biotechnology, and Dr. Shubhro Prakash Hui, who is the head of the Department of Neurosciences, for jointly organizing this one-day event here. So thank you so much. And it's also a big privilege for us to have as a chairperson of this session, Professor Shumantro Chatterjee, who is currently the director of the Center for High Impact Neuroscience and Translational Applications, TCG Crest, Kolkata. He's, of course, a pioneering person in Indian science and needs no introduction. It would be audacious for me to even try and give an introduction to Professor Chatterjee. So we are really thankful to you for making time out for us from his busy schedule. So without any further delay, I would like to invite Professor Chatterjee to kickstart the session.
Good afternoon, and thank you all for being here. I'd like to start by thanking the Indian Academy of Science for having the good taste of inviting Spyros as the Raman Chair Professor, and then creating this opportunity for him to travel across the country from TIFR to NCBS to Calcutta and eventually to Delhi to talk about his exciting work and also in the process inspire younger scientists across the country. As already mentioned by Gitanjali, he's a pioneer in his line of work, especially his work on notch signaling, and he did his PhD at University of Cambridge, which was followed by a postdoc at the University of Basel and also Stanford University. Since 1998, he's been a member of the faculty at the Harvard Med School. And also he's interesting because he straddles both academia and industry. He became the chief scientific officer at Biogen as well. So he has this unique perspective on how science can be done in these two domains. And overall, his research focus has been on the molecular and genetic underpinnings of cell fate determination. And to this end, he uses Drosophila as a model system to understand human biology and neurodegenerative disorders. In 2023, he was elected a member of the National Academy of Sciences of the US. And earlier this year, he received the Gardner International Award along with Iva Greenwald and Gary Strew. And again, thank you very much for being here, Spyros, and please join me in welcoming him to this talk.
Thank you very much. Well, ladies and gentlemen, it's a pleasure and a privilege to be here, and I thank the Academy, and I thank you all for can you hear me or not? You can hear me? No, I thought you, all right, yeah. And of course, Raman himself. It's a privilege to, I don't know if he agrees with what I'm going to say or not, but we'll see, all right? The reception I got in India this time has been overwhelming. A truly warm reception and warm hospitality, for which I really, really, really appreciate. And I'm not just saying that just to say it, I really mean it. The food yesterday was absolutely fantastic. I've learned that Bengali food is better than Southern food. I understand, and that's what everybody tells me. It's been a really, really fun. I've been to Bombay, I've been to Bangalore. I gave, I think, 300 lectures or something like this. But it has been a lot of fun. So today, as opposed to yesterday we solved all the problems, all the philosophical problems of biology. Today I will give you a little tidbit, a small section of biology that has to do with notch signaling, which happens to be my distraction for 40 years, okay? I should also say that the work that I will present, or rather the essence of it, because I will not give you much molecular details and all that. The work that I will present it's in many ways not only our work, but the work of many people, not least of who is my good old buddy Utpal Banerjee, who is sitting right here. That's an honor. I mean, it's a privilege to have you here. So you tell me, you correct whatever I say wrong, all right? Good, all right. So, notch signaling. You can see that this figure here, which was done which was actually created, a Vitruvian man and a Vitruvian fly, in the wishful thinking that, as we said earlier, tell me when I can walk or not walk. I mean, I don't know. I was told I can walk, all right. I was told that the flies, or other humans, are small. Flies are small people without with wings, and that's actually in many ways correct. In many ways correct. All right. So the talk that I will give you, it's divided into. I'll give you a little bit of history. Just came from a session with students, and they were asking the same question: how the hell did I get involved with notch signaling? And I'll tell you about that. I'll talk about the developmental logic of notch. I will say something about receptor and ligands, control of the signals, and then we'll go to some aspects of evolution. Maybe it's my age, maybe it's my stupidity, or my wisdom for that matter, that I'm getting more interested in evolution. Especially since, first of all, I cannot solve it in any way, shape, or form, but it's fun to think about it. And I will also tell you about some proteomic approaches that we have done in an attempt to address some evolutionary questions. Now, everything I will say was not done by me. It was this group of tens of students and grad and postdocs that have contributed to whatever I'll tell you, whatever conclusion I'll make. So, what about notch? Well, notch was uncovered by the man who has solved genetics, and that is Thomas T.H. Morgan, and his magical group of people, of strange and magical group of graduate students and postdocs in Columbia University. And the mutant, the notch, which is called for reasons that I'm sure you appreciate, that is to say there is a wing, loss of function, keep that in mind, we'll come back to that notion. Loss of function will give you notches in the wing. In a homozygous form, this is the fly is dead and has some interesting phenotype we'll go to. But in a heterozygous form, a female, it's an X-linked mutation. And if anything I say sort of genetically sounds exotic, don't worry about it. I mean, if you don't get it, don't worry about it. So it has notches in the wing, and that's why it was called notch. Discovered by 1914 by Dexter, and after that it was forgotten, really. So I'm asked, why on earth did I start working with notch? When I was a graduate student in Cambridge, I started thinking of molecular biology and molecular genetics. And it was at a time where cloning started becoming possible. And you started thinking or hoping to address development in molecular terms. And there were a couple of labs doing that, but not many labs. I read a review of developmental genetics. And at the time, there was only one review. I don't know if you remember that, Utpal, or not. By Ted Wright, a professor in Indiana at the time, and then he moved on. And what he said in this review he said this phrase, okay? He said that if one was asked to choose the single most important genetic variation concerned with the expression of the genome during embryogenesis in Drosophila, the answer would have been to be the notch locus. So like a fish which has a bait in a hook, I swallowed it, the whole damn thing. And 40 years later, here I am, okay? And it did not, it was enough things to do for 40 years, and there's enough space to do for another 40, all right? So, silence for since Dexter, and it was sort of resurrected, if you will, by this wonderful guy, a fantastic embryologist called Don Poulson, who described the embryology of Drosophila in great detail, but also what he was doing, solving a problem in biology, as we said yesterday a bit, that he was collecting mutations, deletions of the X chromosome, and correlating those with specific mutations. A profound thing at the time, because the embryologists did not believe that the genes that affect a wing that has notches, is in any way related to embryonic morphogenesis. And he actually, he was doing his PhD in Caltech with Sturdivant and Dobzhansky, not terribly shabby, and he was looking at those things. So this is a poster that he actually did, and it hangs on my office, and I cherish that. So what I use my finger. Do you have anything? A stick. Utpal, yours is too thick there. No, it's too big. It's too big. Never mind. No, I didn't mean to. Oh, I created a locomotion here. All right, we solved the problem. Thank you very much. Thank you very much. Thank you. How much time I have? Like 2 hours, you said? 1 hour. That's okay. It's okay. It's all right. So, well, you can go away. I mean, you know, or sleep or something. You know, not a problem. So what he did was he narrowed down the notch locus to a particular cytological site, 3C7 for the aficionados, that when you delete that, you kill the embryo. This is now a normal embryo stained for the nervous system, and you can see it's nice and orderly. And if you don't have notch, what happens is that you have a hypertrophied nervous system. But the interesting thing was that this was a hypertrophy of the nervous system at the expense of epidermal structures. So precursor cells have to make a decision, neuroblast or epidermoblast or dermoblast, and this decision is lost, and everything becomes a neuroblast. And this thing grows and dies, of course, right? It has a huge nervous system, and otherwise it's messed up. It's like a tenured Harvard professor or something, okay? So, so that switch was the important thing. However, given that cloning was becoming available, you could actually use this information, 3C7, to try and isolate overlapping clones in a library that had all the genomes sort of in a phage library, and walk from one place to another. Mark Muskiewicz, a colleague who is still a friend, up there on the left is then, further down is now. So he was working on 3C10, a glue protein, for his thesis. So we started from 3C10, walking towards 3C7, isolating overlapping clones, all right? But we were stuck. We were completely stuck. This resolution is not great, but anyway. We were completely stuck on a wall that was created by repetitive sequences. We just couldn't go further, all right? So, to the rescue, another person who has been forgotten now, but if it wasn't for him, I wouldn't be here, all right? And that is, Bill Wilson's. Here we go. Bill Wilson. Bill Wilson was a cytogeneticist. At one point I want to make in Indiana, surrounded by pigs. I mean, Indiana, Ames is surrounded, the campus is surrounded by pig farms. And it smells terribly, terribly. I remember that. So Bill, he was the only person then, the only lab that was working on knots. Nobody else in the whole world. And what he was doing, he was characterizing very, very subtly chromosomal rearrangements around the notch locus, around 3C7. And he had defined a rearrangement, an inversion actually, as it turns out, that allowed us to jump over the repetitive sequences. And that, the rest is history. That allowed us to jump into the locus, isolate the locus, sequence the locus, because without the sequencing, we would not be able to solve terribly much. Before I show you the sequence or the structure of knots, I want to make a few points. The first one is that even though knots is devilishly pleiotropic, it affects everything, as we know now. There's hardly a process that is not affected by knots, all right? The news is if you find something that is not affected by knots, not that it is affected by knots. It affects multiple tissues and developmental events, and indeed, it affects differentiation, proliferation, apoptosis, and movement. All the necessary developmental processes that are necessary to build a three-dimensional organism, all right? I'll come back to that. The developmental logic also is something that, in spite of all this pleiotropy, remains constant. And what it is that you have a layer of cells. One of them decides to become red, or whatever, all right? And the consequences of that decision is transmitted to the next door neighbors, and thus dictating a particular fate for this next door neighbor, or preventing a particular fate, depending on the context. How this is possible? Well, it's possible because the Notch gene encodes a transmembrane receptor which interacts with transmembrane proteins on the surface of two adjacent cells. Now, this is like a ridiculously simplified scheme of Notch signaling, and I'm not going to go into the details of deliciously complicated mechanism of Notch signaling, because I want to talk to you about more general things. I think these are, for a general audience, this is more interesting. I don't want to tell you that this is phosphorylase this or cleaves that. I want to tell you, however, that Notch interacts with the ligands. And what happens, and that's a painful story, is that there is a cleavage that is being initiated by this cell-cell interaction or cell receptor ligand interaction. And the intracellular domain of Notch, presenilin cuts it, and that was a discovery. We call it IVA Greenwald enzyme because it's IVA in worms that discovered that. And this portion, the intracellular portion of Notch, goes inside the nucleus, associates itself with various other goodies, and dictates notch-dependent gene activity, transcription, suppression, and what have you, all right? Now, another thing I need to tell you is that as a rule, abnormal signaling of notch equals abnormal development, or indeed disease. So that signal modulation, how you modulate the signal during development, is absolutely crucial. This is absolutely crucial, how to modulate the signaling. And what I would like to do in the next few slides is talk to you about signal modulation. It will become obvious why I do that, if it's not already, and I want to point out to something, a profound problem in biology. If you look at 3 dimensions, an organism that is built 3-dimensionally, as we sort of mentioned that, alluded to that a bit yesterday, the cell interactions, cell-cell communication is absolutely essential, is absolutely crucial. What has been an amazing surprise is that the signaling framework that makes a fly, makes a fish, makes the president of the Academy of Sciences, Indian Academy of Sciences, or Greek or whatever, it doesn't matter, assuming that this person is human, depends on, which is debatable sometimes, you know. It's not, it depends on a very small number of cellular interaction mechanisms. Now, that is astonishing because it implies that the same signaling framework allows evolution to happen. So novelty, you can invent novelty because you need an arm rather than a wing or whatever, all right, by using exactly the same signaling pathways, which implies, at least intuitively, that it is the interaction between the pathways, the integration between the pathways, the integration between other elements in the cell that is crucial. But the fact that we're using absolutely the same signaling pathways to build 3-dimensional organisms as diverse as flies and humans is a remarkable fact. And in many ways, and I'll come back to that, it's the biggest problem in biology for me, in my head, all right? Now, another point is, of course, we just said that, this is indeed the problem. You know, how do you use the same pathways to get to have evolution happen? So, control of notch signals. I'll give you a few examples to make general points more than anything else. So, the first example I want to give you is the interaction and the modulation of the signal by 2 elements, 2 proteins, deltex and Kurtz. Deltex is a ubiquitin ligase that physically interacts with the intracellular part of notch. And Kurtz, as it happens, is a non-visual beta-arrestin, never mind what that is, but that sticks to deltex, all right? So what happens is that there's a Bengali, an India Sen. What happens is that notch interacts with deltex, as I said, so you can immunoprecipitate, but you can also immunoprecipitate Kurtz only in the presence of deltex, which means that Kurtz interacts with deltex. Now, if you express only Kurtz, where is that thing? Here it is, only Kurtz. If you express only deltex, nothing happens, the wings are okay. If you express both of those together, you get the wing notching. So and we know that wing notching is loss of function, so regulating the signaling. Well, what's happening really in vivo, right? This is a part of a developing wing. This is a single cell layer. And with the magic of genetics, we can take a piece of that. We can create a mosaic that does not have Kurz. The dark means Kurz is green in this particular way, and this does not have Kurz. And if you ask the question, what happens to the Notch protein, you can see the Notch protein is upregulated here. There's more Notch protein in this mosaic. Does this protein signal? Well, a signaling marker. This is now another mosaic, and we know that it's full of Notch protein, but the signal is only in one small bit. So the point I'm making is that it depends on the context whether or not you can actually you can modulate this signal. The signal is modulated in a context-specific manner. So Notch, you have a situation where delta X and Kurz can actually either upregulate or downregulate a signal by modulating the amount of the protein in collaboration with delta X. It doesn't happen by itself, all right? So there's synergy between these molecules, and there's context. That will allow me to see what's happening with signaling. And remember that modulating signaling is important for development and for disease. Think of cancer. If Notch regulation is important for cancer, these kinds of subtle interactions and synergies can be crucial, all right? So what if we take this Kurz and delta X synergy that downregulates Notch and ask the question, can I mutate the genome, all of it or 50% of it as it is in this case, and can I see genes that modulate that phenotype, all right? In other words, it modulates the activity of Notch, delta X, Kurz, one way or the other. And the way we do it genetically, always, is to ask the question, can I find suppressors of that or enhancers? So that's a fourth gene, right? We're talking Notch, it's a fourth gene. And sure enough, we can. You can find both suppressors of that phenotype as well as enhancers, things that make it much worse. How many? In this particular case, we got more than 100 genes that are capable of doing one thing or the other. Now, if you look at ontogeny, if you look what kinds of genes, are they all kinases? Are they all ubiquitinases? Are they all ligases? No heads and tails. We cannot really see any functional kinships from this collection of genes, all right? Now, genetic networks. So remember, this is about more than 100 genes that can actually do that. Already a bit complicated. Now, if we take a loss of function mutation, so if we ask the question, how many genes are capable of modulating Notch signals? And so what we can do is take this loss of function mutation, which we are all familiar with, is the Notch wing notching rather, and ask the question, can I find genes that either enhance or suppress? The answer is yes. We can find both suppressors and enhancers of that particular phenotype. And if we ask the question, if we screen 50% of the genome and ask the question, how many of these can we identify? Well, we can identify more than 500 genes. A bit complicated. A bit complicated, but that's what it is, okay? So let's ask two questions that are connected. How complex is the overall circuitry of Notch signal regulation? And how does Notch integrate its action with other pathways? And I'll give you one example just to give you a sense of the complexity of that integration. Now, E5 and mirror are two of the genes that we found in the previous screen that I just showed you. So now we know that mirror and Notch are capable of interacting with something called a KIT-skewed. Never mind what it is. It's a gene, it's a group of genes that can be up or down regulated by this. We also know that a KIT-skewed are regulated positively by oroquan and capolican, which are negatively regulated by Notch. We also know that wingless can negatively regulate mirror. We know, this is from the literature, we know that TGF-beta can positively regulate mirror. We know that hedgehog can positively regulate mirror. And we know that E5 can positively regulate TGF-beta. So you can see, and that's all I'm trying to emphasize, that this integration is deep and not trivial. Pretty complicated. But that's what it is, all right? So, Notch signal integration, how signals integrate, are very, very relevant for cancer, obviously, all right? So if you take now, we jump in a little bit into the mouse. And if you activate Notch, you activate the receptor, which is simply cutting the intracellular domain and putting it into the nucleus, into the cytoplasm, and then it goes to the nucleus by itself. If you do that, you can actually stimulate proliferation in the crypts of the intestine of the mouse. However, if you knock down wingless by TCF4, those of who are familiar with these molecules, you can actually inactivate wingless, and if you do that, there's no proliferation. So Notch synergizes with wingless to activate, to push proliferation, not without it. Sure enough, in vivo, these are mouse guts. If you look at APC, which is the reason for us doing colonoscopies, really, all right? APC is a negative regulator of winged signaling. And loss of function, if you have APC over plus, if you lose the plus, you get adenocarcinomas, all right? So if a mouse that has APC over plus has about 5 lesions, 5 adenomas, because the APC over plus will make adenomas, not adenocarcinomas. So this has about 5 lesions per gut of a mouse. If, on the other hand, you take APC over plus and add activated Notch there, all hell breaks loose. And you see 115 lesions per gut. And this is quite, as I say, it's quite important because if you look at the Kaplan-Meier curves, the gut, a mouse that has activated Notch is fine after 4 months. APC mouse is dying slowly because it loses heterozygosity. And APC plus Notch goes down precipitously. Within 4 months, it's gone. But what is crucial here to emphasize is that Notch activation by itself is not oncogenic in this context. But Notch activation plus X gives you this kind of adenocarcinomas, all right? And it's lethal. Well, the same thing is happening in the breast. If you take a mouse and you activate activate a notch in the mammary gland, you can actually see this benign, in fact, growths, which are adenomas. These are pregnancy dependent, and again, I will not go into details, but if you go through a few pregnancies like that, essentially then the mice die because they develop adenocarcinomas. And if you look at the cytology, this cytology here is the papillary pattern, which is the benign cytology. And every now and then we can see next to it the solid pattern, which is the typical adenocarcinoma. We presume, but I have not shown that rigorously, that these are accumulation of mutations that would lead to the development of to an adenocarcinoma, right? Now, can we, if Notch activation plus X is cancer, is oncogenic, can we identify X? Can we identify any X's? Well, we are going to do that with flies now. Going back to flies. And if we activate the Notch gene in the eye of the fly, you can actually induce it to become a little bit bigger. There's a bit of a proliferation. And what we can ask again, doing the screen, is can I identify genes that activated Notch plus X, another mutation, can I see proliferation? And indeed, if you activate Notch, you can see a bigger eye. Only Utpal can actually recognize that because that this is bigger. Is it bigger? And you can actually, we can look and combine that with our other genes, and you can clearly see that I can find genes that will blow this eye into an enormous mess of proliferated tissue. In fact, some of these genes are metastatic. You can start seeing eye structures in the abdomen of the fly. How many genes? Where are they? Well, a problem again. There are more than 100 genes again, but if you look at if they are in any kind of functional sort of unit, it's not, they are not. It's all over. There are kinases, everything is there, right? So, I said that there's only one, it used to be only one lab working on Notch, which is the Bill Welson's lab. Now you can count more than 100 labs, I believe, even more. But one directly or exclusively or tangentially are working on Notch signaling because of its pleiotropy, because of its involvement in disease, and so on. So if we take everything, all the genes that have interacting with Notch from Utpal's lab, and my lab, and X lab, and Y lab, and everything together, we call all this, never mind how they are linked with Notch, directly, indirectly, and so on, but they are capable of modulating the signal, because the signal is either modulated directly, but it can be modulated via, God knows where, via Paris or something, all right? So we call this, affectionately, the Notchome, as opposed to the proteome, all right? And if you ask the question how many of these genes are, it is confounding. There's more than 1,500 genes, which is 10% of the genome, really. I mean, that is, in fact, associated with, that's capable of modulating the signal that Notch transmits into a cell. That, of course, can despair even somebody made of marble. I mean, you know, this is like, what the hell do we do now, right? So either you go home and fish and paint and read, go to the Himalayas and with Utpal's Taj, or you try to do something. So what we decided to do was look at protein-protein interactions. I know, so don't tell me, that protein-protein interactions does not mean genetic interactions. I know that. But it's a start to try and deconvolute somehow this mess, this morass of genes that affect not signaling, all right? So what we decided to do, and I take that on me because it's maybe totally stupid, we decided to define the Notch proteome. And the way you do that is you take a few poor students and graduate students and postdocs, you tie them on a bench with, for a long time. Took about almost 10 years to do that. You start tagging cDNA clones. There are about 15,000 genes in flies, more or less, all right? And you start tagging cDNA clones, express these clones into a culture, a cell culture. Fish, use the tag, the bait, to fish whatever else sticks to it, to the protein, and analyze the result by mass spec. I'm not going to complain about the complexity or you know, brag about it, but it was a lot of work, all right? The man who has been instrumental in that is this guy here, Harsha Guru Harsha, an Indian, no less, no more. But he's from Gujarat. I mean, he's not from anything noble like Bengal, but nevertheless, a superb postdoc. And then what you do is you look at these interactions, define complexes as defined by this analysis, I'm emphasizing, all right? And these interactions here are statistical interactions. I mean, we call them interactions because you see them often enough, and you cross-reference them often enough. And there are algorithms that we made, actually, this guy, Julian made, and defined as best as we can protein complexes. Now, protein complexes are important because no one protein acts on its own. It always acts with other things. This reflects one cell line. It reflects, I mean, it has all kinds of confining factors, all right? So, a few things about the complexes. The first version of that was actually also contributed by another Indian. I have to emphasize, brag about my Indians. And there he is. There is this, which I hope you recognize. I don't know. I think he's world famous. I don't know if you know Vijay Raghavan, but there he is. It's all Greek to him, so he messed things up on the first map, and so we had to do it again. No, I'm just joking. So this is now the map, or the hairball, as we call it, that defines about 8,000 individual baits and pull downs. The different colors reflect complexes which are known to have some functional kinship, all right? the interactions are tens of thousands of interactions, all right? Now, a few things about the complexes. This is a blow-up of a particular area of this map. And so we want to find, these complexes will allow us, there are about 600 complexes that describe more or less everything in our system, in our cells, in our this, and our that. So there are several questions that we can ask. Do complexes have a predictive value, first of all? I mean, this starts with a notchome, a protein that has notch that is interacting with notch. Do other complexes, other members of the complex, can I define them to interact with notch? Can I show genetically that these guys interact with notch? And the answer is yes, to absolutely something like 60 degrees almost, 60%, I should say. We find genes that interact. Defining more notchome proteins, as if we need them, and also assigning function to proteins that don't have an assigned function, CGs for the Drosophilists, all right? Now, what happens to a complex when individual complex nodes are disrupted? So here you have a complex, and if I destroy a few of these elements, what happens to the complex? Does it fall apart? Does it stay as it is? And depending on the complex, I'm sure the answers will be different. But if you are interested in a particular function, this is a question that is totally legitimate and necessary. What happens to the network? A more interesting question. So if I completely destroy this complex, what happens to the next door neighbors? Because you can see that this guy is actually connected with other complexes surrounding it. What happens to the first degree neighbors or the second degree neighbors? So you can look at it as a system, if you will, all right? What happens to the network when complex nodes are severed? I'll show you what I mean by that in a second. And what happens to the protein quantities if the quantities are altered? And we can actually now, we're talking earlier with the students, we can actually now do these experiments. We can do isobaric labeling and modulate the activity, the quantity of a particular protein, and see how does that affect signaling, or how does that affect the complex. Quantities is something that we don't like in genetics. We write black or white. Quantity is a pain in the neck. But biology is a pain in the neck, all right? So I'll show you one. And of course, if we use AlphaFold these days to look at three-dimensional aspects of these complexes, how the complexes interact, and thanks to Demis Hassabis, we can actually start doing those things. And they are fun. It's an incredible fun and a waste of time, maybe, I don't know, to play with that, to look at complexes in 3 dimensions and speculate about their ability, about their function. And of course, whether these complexes are conserved. And I'll come back to that, finishing up in the next 3 hours or so, okay? Now, this is a complex that I'll show you one example, all right, what we're interested in. So this is a flotillin complex. This is a snap-snare complex. And what we can see is that synaptobrevin, which is like this guy here, is connecting these 2 complexes. So what happens if we knock out synaptobrevin? What happens to these complexes? Nothing. Something interesting. Something boring? I don't know. We're doing those. Now, conservation, a very crucial aspect of what we do in biology. And thanks to Wade Harper and Steve Gigi, dear colleagues of mine in Harvard, they were interested more, they hate flies. They're not very clever, you know. They don't like flies, but they do like humans for reasons that I don't understand. And so they went ahead and did the same thing with the human proteome. And I'll show you how it looks, and I will go into excruciating detail describing the human proteome. But so it's more complex. But as we were saying earlier, I can't remember when, because I have everything is foggy in my head, when was said what. The quantity of the proteins are different. Usually one fly protein has 4 paralogs in humans. I'm making it extreme simplistically, all right? But that's a good analogy, right? And of course, having these 2 maps, we can, in fact, start looking at complexes and connections that are conserved or are not conserved. And for me, this is extremely interesting and challenging, mind you, at the same time, all right? So is complex conservation a reality or not? We already know that to some degree that's correct, all right? There are some complexes that are conserved across species, like even yeast. Yeast, human, and Drosophila. You can see differences which are interesting. You can see elements that are specific in metazoans versus not. But that's kinds of things that we can actually do. A very important question is whether or not there is a conservation of connections, all right? We said that Notch interacts with proliferation via winglets. And I showed you that this is actually true in mammals as well, all right? But how is there a code of interactions? Like the genetic code, is there an interaction code? So that you start with a basic map of interaction, and as you evolve, you kind of add complexity. So I'll finish with giving you, in other words, I mean, I'm asking whether or not the crosstalk is random, or does it follow some rules? I'm sure it's different between flies and humans and what have you, but are there some basic rules? So I will finish with just giving you a totally speculative, and I'm responsible for this crap myself, so you don't blame anybody else. So the idea is that you have a basic signaling pathways that are crosstalk. So hedgehog talks to wingless, talks to Notch, before talking to TGF-beta, and so on and so forth. And as you go on and develop something more complicated, you add on novelty. A tail, I don't know, a leg or whatever, right? And you go on reaching VJ, again adding more complexity, which is far less complexity than VJ is, mind you, right? This is far less in reality. But if you look underneath, there is a network that defines, that allows novelty to happen. For me, this is the problem. That's the most important problem in biology for me. So this hyper network, I mean, does it exist? Or is it all random and there's no logic to it, which is quite possible. So I am going to stop here. I want to thank you for attending. Thank you for listening. I thank the Raman Chair for giving me this opportunity. I loved the food yesterday. And I want to thank again the people who are truly responsible for my existence, my scientific existence anyway. All right? Thank you very much. And happy to answer questions, of course, or comments, or insults, or whatever.
Comments or questions, and encourage students to start, if possible.
You know, we have been talking with students since this morning, and they have asked a lot of questions. So if you feel like asking something,
Yeah, go
please,
ahead. Anybody.
don't
Utpal.
feel obliged to.
You have to ask a question. Yeah. Which one? You have to ask a question.
He's a student of humanity, so he qualifies as a
Okay,
student.
all right. Let's ask a question. You have the notch, the proteome, all identified as that hairball. Yeah, it almost seems to me, if you want to get to the point of your last slide,
Yeah.
what you need is what genes are excluded from that hairball. What are they doing? I mean, is there anything that is not showing up? That's part of that.
I agree with you. I agree with you, and I think that it's where, as we were saying a bit yesterday, that where AI, this overused and abused word, artificial intelligence, might help, because the complexity is great, and eyeballing things doesn't work anymore. So we want to do that.
So you said 1,500, or I mean, how many genes are in total does one find?
2,000.
2,000. So there's still quite a few left over.
Yeah.
So it is doable.
You're right. This is something which has crossed our brains. The problem now, these days, is that the manpower we have, all right, you know the problem, is tiny. And while Diana Ho, who is still in the lab, and a couple of other people have been trying their best, we have been shifting a bit our interests, and it's moi, who is doing some of that stuff. And that takes forever, all right? Both because of intellectual capacity and everything else.
You're doing
that
great.
I need to do.
You're doing just
exactly.
great.
Yeah.
Thank you.
Thank you, Utpal. But that's, it's a very good point. I mean, this is,
So has there been any spatiotemporal mapping of notch signaling activation? So that may have an answer to the question that it is all random, or there is kind of a...
I'm not sure that I agree with what you say. That is to say, except if we are talking about apples and oranges. I think that, you know, the spatial differentiation, let's say, of notch signaling will not tell you whether this last slide that I showed is true or not. We have to go to, I mean, what Utpal suggested, that's the way to do it at the first level, But, so I'm not sure that this will answer the question. And, you know, people have looked at the notch signaling during development and so on, but, you know, it's not very informative. I mean, it's informative for that particular tissue and so on, right? But it's not really informative. Yeah. Yes?
No, I have basically two questions. One is, since I understand that eye is a good system to look at because it's easier to screen and stuff, but my point is, if you do the same experiment in a different cell,
Yes.
would you
Yes.
expect a different network?
In every single eye, using eye as a phenotype, we always do wing as well. I mean, so in that sense, because if you result that in the eye, you want to make sure that is valid. If you don't look at the eye biology itself, but
Right.
if you look
Right.
at but yeah, you're right. I mean, we're doing that. Yeah. We have been doing it, I mean, traditionally.
And another thing, I may have missed it, I think that was synaptobrevin, the
Yeah.
one in the, so what happened to that experiment? I missed
I don't know, we haven't done it.
Oh, okay, all right. I thought I missed it.
We haven't done it yet.
This is a lovely talk. I'm just wondering, and maybe this is very basic, but Notch signaling usually occurs between cells which are in apposition, like it has to be in contact.
Yes.
So that could be two epithelial cells, it could be an epithelial and a mesenchymal cell, like say during development, depending how the cells are placed. So suppose there is some kind of mutation or something hits and it develops into cancer. Is there a possibility that it might develop into different kinds of cancers in the same organism? Like, could you get an adenocarcinoma and say a sarcoma together?
Adenocarcinoma was just an example I gave. But Notch has been implicated in all kinds of different cancers.
Yeah, but together, simultaneously, is that possible?
Simultaneously?
Because they'll be all genetically similar cells, right?
I cannot think of something specific, but it's possible, I suppose, that you activate it both in the blood and in the mammary gland. It's possible.
Very nice talk. Thank you. Maybe I have missed, but the Notch mutant you have isolated, that's a embryonic lethal, that's mutant. The loss of function of Notch is embryonic lethal.
Is it what?
Embryonic lethal.
Oh, yes, it is embryonic lethal. Sorry about that.
Okay,
I mean,
so you have
my
worked.
Greek ear is sometimes.
No problem. So you have worked with. I'm not sure. So you have worked with hypomorphic allele.
Absolutely, many.
So, and what is the nature of the mutation at the molecular level? I mean, it's a truncated protein. Like, what's the nature of it?
Truncations is a very severe kind of mutation. It can activate or it can kill the receptor. But there are mutations that affect only the eye.
Okay.
There are mutations that are gain-of-function mutations.
Yes, I mean, do you see phenotypes when you overexpress that truncated protein? I mean, do you see any dominant negative effect of that mutation?
Well, not of that mutation, but you can make dominant negative mutations doing other things.
Okay.
Yes, I mean, every single type of mutation you can imagine, we can make. And we have seen.
Yeah.
we have stolen from someone.
Yes.
So
Okay. Thank you.
So, hypothetical question. Hypothetical. So,
hypothetical, yes. I like hypothetical.
I am not the person to ask this, but from my sense, what I am asking you, the complexity, if you keep reducing the complexity, how far can you go? What is the bare minimum requirement for the function, for the system to function? Or the other way around, if you keep on increasing the function complexity, what is the bare minimum requirement?
Well,
the diagram you drew, that in the middle like a bird.
I showed you a slide where it's the bare minimum, the bare molecular minimum. There's a ligand, there is a receptor, there is something that cleaves the ligand, the receptor, and sends the signal into the nucleus.
Right.
And there are some downstream effectors that will control.
What are the interactors do then? If that is the bare minimum, the other interactors which you keep on increasing till the human.
I gave you some example in Deltex and Kurtz, but this is only just one example. There are hundreds of genes that can actually interact, or rather can control Notch signaling. Some of them interact with Notch directly, some of them not.
So I want to take the question from his question. The slide that you showed at the end, where you drew a minimal network and then you drew a hyper network.
Yes.
So the take-home message from whatever you said today is that the core network from Drosophila to human might be conserved, but the hyper network is what evolving and making us what we are. Is that right? Is that what you're trying to say? Okay.
It's a bold
And so the
statement.
core, yeah, to be safe, yeah. And the core network that you're saying, which probably will include flotillins and all the other membrane stabilizing which make the hypercomplexes of any receptor,
Yeah.
those will be the core ones which has to be there. I mean, that machinery will
That's the idea.
stabilize the complex.
That's the idea.
That's the, I mean, that
That's
should
the
remain
idea.
conserved and rest will evolve.
That's
right?
the idea. And
right?
if such hyper networks exist, right, if you don't know, then these would be extremely important for medicine as well.
Yeah, then you can target that.
Because these are key elements that presumably, if you modulate, they can actually modulate disease as well.
Yeah, because your X can be targeted. The machinery cannot be targeted. The plus X will be different for different systems.
Absolutely.
So then you can target X but
Absolutely.
not affect others, right? Yeah.
Absolutely.
So that was not my question. My question is quite different, and probably you did not utter anything about that. My question is, see, when I don't work on Notch, I don't know about Notch. But what we know about Notch is that it's a ductocrine signaling, and then you need a cell-cell interaction, so eukaryotic fate mapping, cell-cell interaction, everything depends on these ductocrine interactions, right?
Yeah.
This is what my basic knowledge is. My question is, you have to cross the barrier of ECM to make this interaction, so always there has to be some, cutting in the ECM to get these cells nearer to have a potential interaction. So isn't it subordinate to some other signaling which actually triggers that cutting so that this juxtacrine signal happens?
Yeah, the extracellular.
Is a question? Okay.
The extracellular domain.
The extracellular matrix. Yeah, because otherwise, the glycocalyx or the ECM, they would be a barrier in any juxtacrine interaction. So what I'm thinking is that you have to make holes, right? You have to make a window to bring delta and Notch together.
If you take a naked cell, if you take a cell that expresses delta on its surface, and you mix it with a cell that expresses Notch.
So it's, I'm thinking about in vivo, the ECM
No,
is there.
I understand what you say, but they stick together.
Yeah.
Now, if the extracellular milieu, the extracellular matrix, plays a role on how close and how fast the ligand and the receptor find each other in order to interact, I cannot tell you anything about it. I don't know.
Okay. So somebody takes a decision, right?
Yeah.
But that's what.
Yeah.
Hi. So I was wondering how Notch is interacting with, let's say, we know every tissue has a terminal differentiator gene. Since you showed us pictures of eye, let's say Walter Gering's PAC6 gene. Is it interacting, or, I mean, what's your take on that?
I didn't understand the question, I'm sorry.
For example, I remember one of Walter Gering's talk where he basically obviously discovered, and then he gave the lecture on PAC6, and even went that far to say if you put PAC6 inside a plant, it's going to grow eyes. So that's too much, obviously.
Well, no, that has nothing to do with Notch.
Yeah. So I was wondering, is there an interaction or crosstalk between, let's say, some terminally differentiated gene and Notch?
I'm not sure what you mean by terminal differentiated gene.
Something like PAC6 or, let's say, antennapedia.
At the end of the day, maybe, yeah, but we haven't really, the connection.
That's the way around.
Yeah.
PAC6 is a very early interacting gene. And Notch, etc. So PAC6 is a universal transcription factor, but it will not make extra eyes unless a certain amount of signal that goes through that region.
Yeah.
So it will make, so what comes first is the signal. Exactly. Now, if it is a wing or an eye or a human being, PAC6, the human being, human mutation, is eyeless as well, and mouse mutation is eyeless. So the signals come in, and then these transcription factors react to that.
Thanks for nice presentation and very really interesting. I have a just quick question about your interaction network. You have a huge network of around 2,000, you said that 2,000 nodes, is it, or it is?
Well, the network.
That big one?
The big one is 8,000 in there.
What, 8,000?
Yeah.
Okay, and you said that when you are destroying some nodes or taking out from the network is, whether the network is going destroyed or like that.
Well, the question is what happens.
Yeah. Okay, so my question is, did you check how many nodes when you are taking out from the network? What is the changes of the clustering coefficient of your whole network? Because it's a huge network of 8,000. And,
No.
Okay, so in that case,
No.
the question may be answered, I don't know, maybe you can check it, that the nodes that you are taking out, within this, whether they're not all like related, any genes are present, number one. Number two, when you are taking out those, what is the changes of the clustering coefficient of the network? So in that case, what is remaining at the last and taking out the list of the taking out the nodes, you can extrapolate the idea. Is it possible?
Everything is possible.
Okay.
But we have not done these things.
Okay, okay.
We have not really done these things. We have not systematically disturbed complexes and observed the consequences. We have not done that.
Technically, these are the driver nodes, I think, what you have taken out or the destroying the, when you are taking out, destroying the network. Yeah. Thank you.
All right, then I think we'll bring this to a close. Thank you again, Spyros, for a wonderful
Thank you.
talk.
Thank you
all for being here. You guys go.
I would now like to request Professor Chatterjee to present Professor Spyros with a small memento. I would now like to request Professor Sanjay Ghosh to present Professor Chatterjee with a small token of appreciation. There you go. Sir, apne. Once again, thank you all for tea and refreshments are outside for all of you. So please enjoy.